Hello class, this is Dr. Anderson and these are a series of presentations on the sensory system, and we're going to start with the receptors. Here are our learning objectives. First of all, there are a lot of terms that I need you to be aware of, and you'll need to know about those terms just to understand the other material. So sensory transduction, labeled line principle, receptive field, absolute sensory threshold, Weber's law, adaptation, tonic receptors, phasic receptors, two-point discrimination, temporal and spatial summation. Alrighty, so we're going to talk about receptors and what are the properties of a receptor potential, also called a generator potential, and compare and contrast that to an action potential. Given that action potentials are all or none, how is a sensory pathway going to convey the idea of intensity to the central nervous system? What is a sensory threshold? And what's happening in the receptor and the sensory neuron aligned with it below the sensory threshold and above the sensory threshold? We need to know what sensory modalities are. Those are basically different kinds of sensations and how those are conveyed to the CNS. We're going to talk about the size of a receptive field, how that influences two-point discrimination. We need to compare, say, a highly discriminating part of the body like the fingertip and a less discriminating part like on the shoulder blade in terms of two-point discrimination and with the basic classifications of neurons. So this is a picture from your textbook. This is a sensory neuron. Here are the terminals that are ending up in the nervous system. Here are the ends, the nerve endings, or the primary receptors out here in the skin, and some kind of stimulus, when it touches this area of skin, will activate this receptor. So the area of skin that's innervated by this receptor is called the receptive field. About here, we're going to see voltage-gated sodium channels. So this is the axon that allows us to have an action potential. This cell body is going to be in the dorsal root ganglion. Notice nothing is going to synapse on it. It doesn't have any processes. Nobody talks to this guy. This is the cell body that keeps this neuron alive. Then the action potentials go into the central nervous system, and out here, neurotransmitter is going to be released. So modality are the different kinds of sensations. So a mechanoreceptor can sense pressure and vibration and touch and that sort of thing. Thermoreceptor is going to sense hot or cold. A nociceptor is for pain. We have electromagnetic receptors such as those in the eyes, the special senses. We have all kinds of chemoreceptors in the visceral organs. There are basically 4 somatic modalities. We have touch, we have temperature, we have pain, and we have proprioception. Here are some of the touch receptors you might see in the skin. So here are what we'd call free nerve endings or uncapsulated receptors. That could be a nociceptor, could be a temperature receptor. It depends. Here are some encapsulated receptors. We might get some temperature with some of these guys. These are probably a lot for touch. This is a pretty famous receptor called a Pacinian corpuscle for vibration and deep touch, and we'll talk a little more about that one. So I don't expect you to memorize the names of all the receptors. Just realize that different receptors allow for different modalities, and we'll talk a bit more about that. So as I said before, in the somatic nervous system out in the body, we have touch, temperature, pain, and proprioception in terms of our modalities. And these sensory receptors perform a function called transduction, basically converting one kind of signal energy to another kind of signal energy. So pain energy gets converted to an electrical signal. Touch or a mechanical signal gets converted to an electrical signal. and so forth. So transduction is a general word. Those of you that work in the clinical arena, you have a pressure transducer. It's taking a pressure signal and turning it into an electrical signal you can see on your display. This is the very same kind of thing. The receptor is performing transduction, and they're doing it with transduction channels. So these are channels that respond to the particular modality. So maybe pain could cause an intense pinching or ischemia or release chemicals. Whatever those things are, they would tell these receptors to open their channels. This would depolarize the receptor, and that's converted into an electrical signal, and that's got to be converted then into an action potential to go to the central nervous system. So the receptor field, that's the area of skin that's monitored by a particular receptor. So we have 3 receptors. Notice there's a little bit of overlap, and when we stimulate in the middle of the central receptor, we get a nice big signal and a lesser signal on either side. Receptor fields vary in size, they vary in overlap. If you want to have a higher resolution, you need lots of closely packed receptors. We'll talk about that a bit in further slides. If you get a stimulus here, we're going to generate a graded potential that's going to be converted to action potentials. So notice where the stimulus is the greatest in the center. If we look at which afferent neuron compared to action potential frequency, the greatest stimulus corresponds to the greatest action potential frequency. This neuron is going to be connected to another neuron that's going to be connected to another neuron that goes to a particular spot in the brain. And action potentials generated from this stimulus are always going to travel along that pathway. That's part of how the brain's going to know what the stimulus is and where it came from. And so the sensory nervous system follows the labeled line theory, that there's a point out in the periphery that corresponds to a particular point in the brain. And this labeled line is parallel to that labeled line, and that labeled line is parallel to that labeled line. So people also call this parallel processing. The absolute sensory threshold is something that comes from both psychology and neurophysiology. Basically, you have a subject, you expose them to a stimulus, and what's the lowest intensity stimulus that they can detect 50% of the time? That's your absolute sensory threshold. Would that change if the person had 14 cups of coffee? It might. So it's not necessarily a fixed point in time or a fixed sensation, but typically when neuron recordings are made, when the person says, "I feel the sensation," coincides with sensory neurons sending action potentials back to the CNS. Sometimes sensory threshold can change because of things going on in the periphery or things going on in the CNS. So for example, someone having a lot of caffeine or someone being very tired or that sort of thing. When a stimulus of the right modality touches a receptor, you get a receptor potential or a generator potential, which is a kind of graded potential. So a cold stimulus touches a cold receptor, and you get a receptor potential or a graded potential. If that reaches threshold, then there will be action potentials in the neuron going towards the central nervous system. Again, here's a Pacinian corpuscle. If we have a pressure stimulus that activates the transduction channels in this receptor and we reach threshold, then action potentials will go to the central nervous system. We're not going to talk a lot about special senses, but for example, a sugar molecule touches a sweet receptor, that releases some neurotransmitter, and then if we bring this neuron to threshold, then a frequency of action potentials will be sent to the central nervous system. Now notice, all have different modalities: cold, pressure, chemical, but they all eventually are translated to a graded potential, which, if it reaches threshold, corresponds to a frequency of action potentials in a sensory afferent. So how do we convey intensity? So here's our receptor with its transduction channels, and here's the stimulus of the appropriate modality. We stimulate the receptor, we get a receptor potential, but we have not reached the threshold of the voltage-gated channels in the initial segment. And I think of the first segment here, on this diagram, they have it here. But wherever the voltage-gated sodium channels are, that's where it's going to start. We're below threshold, so we have no action potentials. We have no action potentials going to the axon terminus, we have no calcium influx, and we have no neurotransmitter release. If we increase the intensity of the stimulus, then we've got a bigger graded potential, which leads to some frequency of action potentials, because now we're above threshold, and those action potentials arrive at the axon terminus, and we get some release of neurotransmitter. If we increase the intensity of the stimulus, then we get an even bigger receptor potential, which leads to a higher frequency of action potentials. That higher frequency of action potentials is preserved in the axon terminus, or down the more distal end of the, well, and actually in this case the proximal end of the neuron, and then we get more neurotransmitter release. So this is how we're conveying intensity. More intensity of a stimulus means a bigger receptor potential. That means a higher frequency of action potentials and a bigger release of neurotransmitter at the synapse. So intensity is encoded by frequency. As we increase the intensity of the stimulus, we get a bigger receptor potential and the frequency of action potential increases. Now, action potential frequency may not stay stable even if the stimulus is in place. There may be some changes in that receptor. And those changes in the receptor then cause less activation of those transduction channels, and we could get a slowing of the action potential, or the action potential frequency. So we are still getting a report of action potentials, but the frequency at the onset is greater than the frequency at the offset. So we see some adaptation. This is a very slowly adapting receptor. So we can categorize receptors in terms of how fast they adapt. Phasic receptors, I think, are going through a phase. At the onset of the stimulus, we have a frequency of action potentials, and we get a rapid adaptation, and they drop off. And we get no report until the stimulus is removed. So these kinds of receptors are very, very good at saying when a stimulus goes on and goes off. So they're good about changes in stimuli or the rate of a stimulus. So if you have something going on and off and on and off, that kind of thing is called rate coding. Slowly adapting receptors are tonic. They are showing tone. So there's a little bit of adaptation, but a constant report of action potential. So the CNS is always kept up to date about what's going on in the periphery. So something like a nociceptor should not adapt, because you need to know there's pain out there so you can take care of yourself. Now, how do we discriminate intensity? How do we know if this stimulus is more intense than the other stimulus? Well, we have something called Weber's law to help us describe that. Discrimination is best at low intensities. So I may hold my hand out, and my lab partner puts a little weight on my hand. I notice that it's there. Then they put a little heavier weight. I can tell, yeah, it's a heavier weight. So they put on 1 gram, then they put on 2 grams, then they put on 3 grams. I can tell the intensity. But then they put on 4 grams, and I'm not really sure. If they put on 10 grams, oh yeah, now I can see it's an intenser stimulus. But if they put on 11 grams, I can't really tell between 11 grams and 10 grams. But if they put on 20 grams, yeah, I can tell. So as the stimulus gets bigger and bigger, I need bigger changes in order to notice that the stimulus is more intense. So that's what Weber's law describes. How do we discriminate resolution or points? So we have 2-point discrimination. So notice we've got some receptor fields that are large. There's some overlap, but they're very large. Then they have their afferent neuron going with them. There's the cell body in the dorsal root ganglion. Its synapses in the spinal cord somewhere. Notice I've got something, this is a caliper, and the points are a certain amount apart. These 2 points go in the same receptor field. We're only going to activate this receptor. We are only going to activate this pathway. So as far as your brain knows, that's just 1 point. How are we going to tell if there's 2 points? Well, other parts of the body, say like your fingertip, have smaller receptive fields and more of them, and they're all closely packed. So notice my little sensory caliper. The points are about the same distance, but I'm able to excite 2 receptors. I'm able to activate 2 parallel pathways. So these go along the labeled line. There are going to be 2 points in my brain that indicate there were 2 points out in the periphery. So if this is the back of your neck and this is your fingertip, I have better 2-point discrimination in my fingertip because the receptor fields are smaller and more closely packed, and I have more resolution. It's like having more pixels in a camera so that you can see more detail. Now, on the back of my neck or on my back, I just need to know the stimulus is there. I don't exactly need to know exactly where it is. So I don't want to waste a lot of energy on a lot of detail. I don't read Braille with my back. I would read Braille with my fingertips because that's where my 2-point discrimination is. Alrighty. We have talked about how sensory neurons behave. In the next presentation, I'm going to talk about how those receptors work together in a pathway so the periphery can talk to your brain. So I'll see you in the next presentation.